Welcome to The Explainer. Today, we're diving headfirst into one of the absolute most exciting medical frontiers in all of regenerative science. We're talking about the human eye and how breakthroughs in modern ophthalmology are merging genetics, cell biology, and regenerative medicine to completely transform how we understand and treat ocular disease. Now, I know it sounds straight out of a sci-fi movie, right? But I really want you to consider this question for a second. What if we could actually regrow human vision? The ultimate stakes here are incredibly profound. We're talking about literally replacing damaged retinal tissue to cure blindness and restore sight. This isn't just about slowing down a disease anymore, it's about reversing it. Section 1. The Regenerative Ocular Frontier. Okay, let's dive into this. Because to understand how we're going to achieve this massive goal, we first need to understand why the eye is the absolute perfect place to start. Well, unlike deep internal organs, the eye is incredibly accessible. Doctors can look directly into it. Because it's so easy to monitor using direct imaging tools like OCT and fundus photography, the eye serves as the ultimate testing ground for regenerative medicine. We can deliver targeted treatments exactly where they're needed and watch their progress in real time. Plus, the eye is relatively immune privileged. That basically means it has a somewhat different relationship with your body's immune system, making it a whole lot more forgiving when we introduce new therapies. Section 2. Pluripotent stem cells explained. So what actually is a stem cell? Think of it as a biological blank slate. It's an undifferentiated cell that comes with two true superpower properties. First up, self-renewal. It can continuously divide and literally just make more of itself. And second, differentiation. It can transform into highly specialized tissues. In ophthalmology, researchers want to use these magic building blocks to generate fresh retinal pigment epithelium, photoreceptors, and ganglion cells to replace the ones a patient has lost. Now, what's really interesting here is the contrast between the two main types of pluripotent stem cells we use. On one hand, we have embryonic stem cells. These are derived from early embryos, so they naturally possess incredibly extensive developmental potential. But on the other hand, we have the modern miracle of induced pluripotent stem cells, or iPSCs. These are adult somatic cells, just regular cells from a grown person that scientists have genetically reprogrammed all the way back into a pluripotent blank slate state. And that brilliantly illustrates why iPSCs are such a massive breakthrough. It's basically cellular time travel. We can take mature cells, like skin fibroblasts, right off your arm, and literally teach these old adult cells new embryonic tricks. They completely regain that early developmental capability. That means we don't necessarily have to rely on embryonic tissue to get the stem cells we desperately need. So how do we actually get these blank slate cells to become part of a working eye? Let's walk through this cellular recipe together. Scientists use a process called embryological mimicry. Step 1. You start with your pluripotent stem cell. Step 2. You guide it using highly specific signaling pathways and growth factor cascades. Specifically, we're talking about the WNT, sonic hedgehog, and notch pathways. By mimicking early eye development right there in the lab, we hit step 3. The stem cell matures perfectly into functional retinal tissue, like retinal pigment epithelium or photoreceptors. Section 3. Treating retinal disease. Let's move from the lab to the actual battlefield of human ocular diseases. One of our absolute primary targets right now is age-related macular degeneration, or AMD. In AMD, the retinal pigment epithelium degenerates, and that causes a secondary loss of photoreceptors, the cells that actually capture light. The clinical strategy for this is just fascinating. Researchers are transplanting specialized stem cell-derived retinal pigment epithelial cells right beneath the retina. The goal here isn't just to put a Band-Aid on it, it's to support the retina's metabolism, preserve the surviving photoreceptors, and actually improve visual function. And the best part? Early clinical trials are already showing that this is biologically feasible. But what about inherited retinal diseases, like retinitis pigmentosa? Well, for that, scientists are using this incredible concept called retinal organoids. These are literally miniature, laboratory-grown retinal tissues derived from those pluripotent stem cells we talked about. These mini retinas allow researchers to safely study the precise genetic mechanisms of a disease right there in a petri dish, so they can test out gene-corrected therapies before they ever touch a human patient. Now, while a lot of that retinal therapy is still largely experimental, I want to emphasize that stem cell use in ophthalmology is already an established clinical reality for the surface of the eye. When a patient's cornea is damaged by severe triggers like chemical injuries, Stevens-Johnson syndrome, or ocular cicatricial pemphigoid, they can suffer from limbal stem cell deficiency. To fix this, doctors are literally transplanting limbal stem cells from the healthy corneoscleral junction to restore the structural integrity and function of the eye's surface. We're already doing this today. Section 4. Roadblocks to regeneration. Okay, we have to talk about the roadblocks, because despite the massive potential, this is definitely not a silver bullet yet. Let's map out the terrifying risks, but also the brilliant solutions being developed to solve them. First major challenge, tumor formation. Undifferentiated cells can proliferate uncontrollably into multi-tissue tumors called teratomas, which is obviously really bad. The solution? Researchers have to ensure absolute complete cell differentiation before transplantation. Challenge 2 is immune rejection. To stop the body from attacking the new cells, we can use immunosuppressive therapy, or even better, autologous iPSCs, which basically means making the cells directly from the patient's own body. And finally, ethical concerns regarding embryonic tissue are largely being bypassed altogether by utilizing that amazing iPSC technology. So the crucial point is this. Simply restoring the anatomical structure and replacing some cells isn't nearly enough. The cells must functionally integrate. Let's walk through this specific hurdle. First, the transplanted cells have to achieve long-term survival. Second, they have to physically align in the appropriate orientation inside the eye. Third, and this is by far the hardest part, they must literally wire themselves into our central nervous system by forming synaptic connections. And finally, they have to achieve functional signal transmission, because if those brand new photoreceptors can't actually send a light signal to the brain, they're not going to restore any vision. Section 5. Genetics and future therapies. Just consider the sheer scale of diseases that modern molecular genetics has identified for potential gene therapy. We're looking at things like retinitis pigmentosa, Stargardt disease, Leber congenital amaurosis, congenital glaucoma, and retinoblastoma. Because the Human Genome Project expanded our understanding so deeply, scientists can now pinpoint the exact mutated genes causing these progressive degenerations. And that opens the door for gene-corrected autologous iPSC therapy. We're talking about fixing a patient's own cells at the genetic level before putting them back into their eye. But let's connect all this dense science back to the human element. For a patient dealing with visual loss, understanding the underlying genetics is vital. Genetic counseling has become a massively important field in ophthalmology today. By mapping out Mendelian inheritance patterns, figuring out if it's autosomal dominant, recessive, X-linked, mitochondrial, counselors can help families truly understand their specific disease prognosis, their recurrence risks, and their options for these incredible future therapies. And that brings us back to our ultimate question, summarizing the true promise of regenerative medicine. Are we entering an era where we don't just preserve vision, but regenerate it? As we combine genetic diagnosis, gene therapy, and stem cell engineering, we are literally paving the way for personalized regenerative strategies that could make blindness a thing of the past. The science is moving incredibly fast, and the future of ophthalmology is looking brighter than ever. I hope this explainer helped clarify the breathtaking science of ocular regeneration and leaves you eager to learn more about the future of human health.